Hey everyone, welcome back to Cells to Cell. Today we're actually doing Trivia Tuesday, and we're going to be doing the 9-question quiz that goes over cell and gene therapy. So, starting off, Nick, how you doing?
Hanging in there.
All right.
I'm hungry.
Yeah.
That's okay. I'm hungry for knowledge,
Ooh,
to say the least. So.
nice.
All right. You get the first question.
All right.
All right. Question number 1. 6 CAR T cell therapies have been approved by the U.S. Food and Drug Administration. Which cancer do most of them target?
Okay.
Is it acute myeloid leukemia, lymphoma, multiple myeloma, or breast cancer?
So there has been no current treatment for AML, acute myeloid. Multiple myeloma, there's one CAR T, and I think Bristol Myers Squibb does it. Breast cancer, I don't think there's been CAR T approved for it, so I'm going to say lymphoma, because 6 of them should be lymphoma.
Yeah, that is correct. 4 of the 6 CAR T therapies, I couldn't think of the word, are designed for different forms of lymphoma.
Nice.
So,
Next question, Nick. Speaking of CAR T therapy, the phrase chimeric antigen receptor that makes up the acronym CAR is called such because the genetically engineered receptors are artificial. The word chimera is also a term from Greek mythology that means what? Dragon, magician, monster, or chameleon? Ooh.
So I don't have as much of a CAR T background as you do.
But this is about Greek mythology, dude.
This is about Greek mythology. I'm going to go with chameleon because CAR Ts are taken out of the body, run through a solution, and they're given new cell surface proteins. And I'm assuming that putting them back in allows them to kind of camouflage themselves and get into places that need to be healed or get into cancers without being noticed by those microenvironments and getting attacked by the immune system. So
that is great reasoning. But they don't go through a solution, they just get genetically engineered, but okay.
Is it? But like the thought was there.
The thought was there. Chameleon is correct.
Sweet.
Oh no, sorry, the correct answer is monster.
What?
It's monster. Okay. In Greek mythology, a chimera is a monster that has a lion's head, a goat's body, and a serpent's tail, and also breathes fire. Oh, okay, so I guess CAR T is because it's made up a lot of different parts.
Okay. I mean,
Weird question.
I think they should change it because of my reasoning, but that's just me. Okay.
Agreed.
In recent years, researchers from China and the U.S. have genetically modified a patient's T cells to better see and kill cancer using what 21st century tool? Is it A, RNAi?
No.
Is it CRISPR? Is it, I'm going to assume that's zinc fingers, but ZFNs? I don't know what ZFNs are.
Okay.
Or talons.
CRISPR is to genetically modify something. RNAi is RNA interference.
Yeah.
Zinc fingers or ZFI and ZFNs, I have no idea what that is. And talons, I feel like I might have heard that, so I'm going to just go with talons.
I don't know the answers. I'm going to go with CRISPR. If ZFNs is zinc fingers were used before the 21st century for genetic modification, whereas CRISPR was discovered in the 2000s, I believe. I think it was, or maybe it was discovered. I know CRISPR was discovered after zinc fingers.
Okay, but
I've never heard of talons.
Yeah, same.
So I'm going to go with CRISPR. You're going to go with talons. CRISPR's correct.
CRISPR's correct.
Yes. Chinese trial involved a man with lung cancer, and the U.S. trial involved patients with metastatic sarcoma and multiple myeloma. Initial findings from both suggest that the treatment is safe, but more work is needed.
Right.
I'm going to look up really quickly
CRISPR.
I'm going to look up what ZFNs are. Pros and cons of, okay. Yeah, there's zinc fingers. Zinc finger proteins. Zinc finger proteins, yeah.
Oh, okay. It's like a really old
It's
form
an old
of
form of CRISPR. is
of
what it
CRISPR?
is.
Interesting though.
Mm-hmm.
All right, Nick. In gene therapy, a normal gene to replace a faulty gene is directly introduced into a patient's cells. True or false? So in gene therapy, a normal gene to replace a faulty gene is directly introduced into a patient's cells.
I'm going to go with false.
I feel like that's also false.
Yeah.
That's
I think
correct.
directly introducing a gene would be like suicide for the gene.
Yeah.
Because it would have no protection. Even DNA and genes floating outside of the nucleus where the DNA is held, they have their own protective measures so they don't get eaten up or destroyed by enzymes that, monitor the cell in general. Because, lysosomes are something that go around and literally eat up debris in cells, and they could pick up DNA and see them as debris. So if you just throw a regular gene in there, it's going to get destroyed.
Yeah. The answer says you need some sort of viral vector, so
Yeah.
like literally like a platform that can carry the gene into it. And if you watch one of our episodes with Ryan, episode number 3, he goes over how nanoparticles are the delivery vessel for these genes.
Yeah. Nanomedicine's incredible. Okay, next question. In 2012, 6-year-old Emma Whitehead was the first to receive a CAR T-cell therapy that turned her leukemia prognosis around. Which virus did researchers at UPenn use to deliver Emma's cell therapy? Was it HIV, an adenovirus, the influenza virus, or the cytomegalovirus?
2012, 6 years old, CAR T. I don't want to say HIV. I don't want to say influenza. I'm going to go with adenovirus. I'm the most familiar with adenovirus.
I would say adenovirus too.
But if it's something novel, it might be HIV.
It's not. I don't think it's
not?
HIV. No.
Okay, either influenza virus or adenovirus. So adeno. We'll go with adeno.
The correct answer is HIV.
It is HIV.
Interesting. To perform the treatment, doctors removed millions of Emily's T cells, a type of white blood cell, and inserted new genes that enable the T cells to kill cancer cells. The technique employed a disabled form of HIV because it is very good at carrying genetic material into T cells, which makes sense. Usually, this goes back to my undergrad and my virology days, but that reminds me that when we're talking about viral vectors, usually you're going to choose the capsule that most easily attaches to or binds to the tissue you want. So when we are talking about, let's take COVID for example, right? You're going to want to, if you're going to want to insert a gene, you're going to want to, let's say, use the influenza virus or an adenovirus that's going to easily get into the lungs and easily be able to get into those cells and deliver those genes, rather than HIV, which would circulate throughout the body and doesn't really get lodged in the lungs. So when you're trying to attack immune cells, you're going to want a virus that attacks immune cells. So that makes sense.
But they say they removed millions of her T cells. So that means they took them out.
Yeah.
And then to make sure each and every one of those T cells got genetic material, they gave her a disabled version of HIV, which is interesting because there's actually a researcher here at Case that's working on using disabled HIV to send genes also. So
Yeah,
it's
very
very
cool.
interesting.
All right, next question.
Okay, so in 2010, regulators approved a therapeutic cancer vaccine that trains the body's own immune system to fight the disease. What disease does the therapeutic vaccine target? Acute myeloid leukemia, breast cancer, prostate cancer, or multiple myeloma? So a therapeutic vaccine
Cancer vaccine?
that trains your body's own immune system to fight the disease. I think I know the answer to this one, but I did not know it was in 2010.
I'm going to go with multiple myeloma.
Okay.
Is that wrong?
I think it's breast cancer.
Really?
I think so, but I thought that was something recently. Oh, it's prostate. We're both wrong.
Jeez.
The vaccine called Proveng is personalized for each prostate cancer patient and was the first one of the cell and gene therapies approved by the FDA.
That's crazy. That's really cool.
Wow.
And one thing that people are searching for is that kind of personalized medicine. So I guess they have it for prostate cancer now, which is really cool. All right, next question.
We're doing horrible.
This
on
is a hard quiz.
Yeah.
All right. In 1953, scientists discovered the first double helix, the twisted ladder structure of the DNA that gives rise to modern molecular biology. Where was this discovered? First off, do you know who discovered it?
Watson and Crick.
Yeah.
And those names are so British. So I'm going to say UK. Watson
Yeah.
should be UK.
Yep.
That's correct.
Yep,
The
it was
other
in
options were
Cambridge.
France, US, or Netherlands. We went with the UK.
Who was the lady that did all the X-ray crystallography for him?
Marie Curie, I know, did a lot of chemistry. And she's a
No.
very pronounced woman figure.
Rosalind Franklin. Yes. She did all the X-ray crystallography that basically gave them the scaffold to build a double helix. So,
Cool.
look, women in science,
Make it
they are
big
they
moves.
make it happen too. All right, next question.
In 2003, the National Medical Products Administration, formerly the China Food and Drug Administration, approved the first commercially available gene therapy to treat what form of skin cancer? Melanoma, basal cell carcinoma, squamous cell carcinoma, or Merkel cell cancer? This is so difficult,
Yeah.
but we should, I guess, be familiar with this because these are all apparently approved treatments of gene therapy.
I know since then there are ways of curing melanoma even after it's metastasized. But that was using stem cells and not gene therapy. So I don't know.
I don't think it's squamous cell. I'll give you that.
I'm going to go with basal cell carcinoma. I think that's a safe bet. The correct
Oh.
answer is squamous cell carcinoma.
Oh,
Talk me
God.
out of it.
Oh, God.
Talk me out of it.
Sorry.
It's okay.
Sold under the brand named Gendicine, the world's first commercial gene therapy. Targets head and neck squamous cell carcinoma. That's so fascinating because I worked in a lab
Yeah.
focused on squamous cell carcinoma. So
The virus is engineered to carry the gene that has instructions for making tumor-fighting proteins. Interesting. Good. Good. We're getting closer and closer to cures for cancer, which is awesome. All right. Next question. Researchers are developing off-the-shelf allogenic T-cell therapies derived from unrelated donors that could potentially treat hundreds or thousands of patients. What is the major hurdle of these allogenic therapies? Graft versus host disease, cost, production delays, cytokine release syndrome. Graft versus host disease?
Yeah.
Yeah.
Cost and production delays are mundane things
Compared to...
compared to something like allogenic. Allogenic meaning it comes from somebody.
Yeah.
Autologous comes from your own self. So having pretty much like an off-the-shelf cell therapy is unheard of. Because if you're giving some live cells to somebody else, the biggest thing is graft versus host, meaning that their cells won't be compatible with yours, your cells won't be compatible with theirs. So
Right.
having an off-the-shelf one is wild. Wild.
That's what a company called Athersys here in Cleveland is trying to do, is make an off-the-shelf stem cell therapy called Multistem. Just a fun fact. We'll probably talk about Athersys a little bit more.
University of Pennsylvania announced this year game-changing findings from its CAR-T research that will likely have a major impact on future drug development. What did they reveal? That it had developed the first ever CAR-T for lung cancer. That's the first time its CAR T was able to cross the blood-brain barrier, that 2 of its first patients, dosed 10 years earlier, were cured of cancer, that it could mass produce the T cells needed for research, opening the door to wider applications of CAR T. This is a little unfair because Nick hasn't gotten his education yet on CAR Ts, and I have. But But the question is asking, will likely have a major impact on future drug development. So which one of these has implications for further research? Because first ever CAR T for lung cancer is kind of specific. I think
I don't know why I want to go with that one. D.
being able to mass produce.
Yeah.
So yeah, either I was thinking that one or the blood-brain barrier one.
Yeah.
Because it would impact future. But if you're talking about the broader spectrum, I'd say mass produce because that's
No, patients were
Ooh.
cured.
patients were cured is the answer.
And they posted in Nature. Nice.
So the CAR Ts infused in the patients remained detectable at least a decade after infusion.
That's wild.
So that means those engineered T cells didn't just.
Go in and die off. They stayed in
Yeah.
and fought, which is awesome.
So every time that cancer even came back remotely, it was fought off, and that's curing, is it not?
Yeah, that's super cool.
And that's all for this quiz. We got a 4 out of
We got
10.
40%. But some of those questions were really hard. I
That's
did
a
not
little-
expect that quiz to be that hard. But, like the point of this is not for us to sit here and
Act like we
act
know
like
everything.
we know everything.
No.
We're still learning. We're still growing and trying to figure out this field just as much as everybody else. So, all right. Well, that was this, Trivia Tuesday, which wasn't so successful, but we did learn a lot, and I feel like we got to talk about some good points. So, Heem, you got anything else before we kick it off? All right. Well, I'm Nick. That was Heem, and thanks for watching Cells to Sell. We'll see you next time.
See you guys.